Welcome to LankanTutor Biology 2.4. If you're not watching this on LankanTutor.com, we suggest you do so, and let's get into the lesson. So we left off after we finished carbohydrate in organic molecules. Now remember what an organic molecule is. Carbohydrates, proteins, lipids, and nucleic acids. Since we finished everything we need to know about carbohydrates, we're going into proteins today, and hopefully we can cover that out. Now, what exactly is a protein? A protein is a linear polymer. Now, you need to remember this. It is a linear polymer. It is not a branch structure. It's made out of subunits called amino acids, and they have high molecular weights. If you remember, in organic molecules, we divided molecules into two parts, macromolecules and micromolecules. It is considered a macromolecule if it has a molecular weight of 10 to the power 4 to 10 to the power 10. Proteins are all macromolecules. If you remember carbohydrates, the organic composition was carbon, hydrogen, and oxygen in the formula of CxH2O(Y). In proteins, they contain carbon, hydrogen, oxygen, and it also has nitrogen and sometimes sulfur. Amino acids are the monomers of proteins. Although they have several amino acids in the body, there are only about 20 of them which join to form proteins. So amino acids contain a basic NH2 group and a carboxyl group, which means it's a COOH. Don't worry, you'll learn more about these groups in chemistry. You don't really need to go into detail here. It's just that you need to remember that they have it. So because of this, amino acids have both basic and acidic properties. Therefore, it's called an amphoteric molecule. Since there are about 20 different amino acids, I'll just say the most simple form of amino acid, it's called glycine. The diagram I'm drawing right now is a picture of glycine. And on the right here, we have the general structure of an amino acid. An R group is any CHO group, and it has a carboxyl group, an NH2 group, and a carbon atom bonded to a hydrogen atom. Two amino acids can combine to form a dipeptide. The dipeptide linkages are formed between the bond of the carboxyl groups of the amino acids. If more and more amino acids are joined, it's called a polypeptide chain. Now remember that the proteins are not branch molecules, they are extremely long chains, depending on what protein it is. A protein can consist of one or more polypeptide chains, and they are polymers made up of amino acids. Not all proteins look the same, the structures are different. Proteins get classified into 4 categories depending on the shape. The first one is called a primary protein. The reason that it's called a primary protein is because it's a linear structure which has no bending or folding. It's just a sequence of amino acids in a polypeptide chain. That is as simple as it gets. And then we go on to the more complicated types of proteins. So the secondary structure is a bit more complicated, and it has 2 examples that we study about. The first one is called an alpha helix structure or a helical structure. A helix is generally a coil, a strand that goes around and around each other, and it is joined together. These bonds between the two circles, or the two vertexes, not exactly vertexes since it's a circle, the edges are joined together with hydrogen bonds. And the other type of secondary structure is called a beta pleated structure. The alpha helix and the beta pleated. The pleated is just a chain which is pleated along the structure, and again they're all joined together by hydrogen bonds. An example for these secondary structures would be silk and the cobwebs that spiders produce. They're all secondary proteins. Next we move on to tertiary proteins. Now tertiary proteins are a bit more complicated. It actually has a definition that you need to remember. It is the bending and folding of polypeptide chains to produce a globular shape. it has lots of bonds. Namely, they have disulfide bonds, ionic bonds, hydrogen bonds, and hydrophobic bonds. Now you need to remember that in these bonds, the strongest bonds are the disulfide bonds, and the hydrogen bonds can easily be broken down with heat and physical pressure. And another thing you need to remember is that all the enzymes are tertiary proteins. Now again, let's go back to tertiary proteins. You need to remember bending, folding, polypeptide chain, and globular shape. If it doesn't have a globular shape, it is not a tertiary protein. If it has a globular shape, it could be a tertiary protein or it could be a quaternary protein because it can get more complicated. So, just like tertiary proteins, you're supposed to give a definition of quaternary proteins. It is the aggregation of two or more polypeptide chains to form a complex structure. Now remember that all tertiary proteins have globular shapes, and for it to be considered a quaternary protein, it has to have more than two globular shapes, and it has to be joined together, and it can also include other groups. For example, hemoglobin, which consists of 4 polypeptide chains, and 2 distinct types of alpha and beta, and also a non-proteinous heme group. So you can see that even though it has the polypeptide chains made from amino acids, it can also have non-proteinous structures like heme. Another example for a quaternary structure is collagen. The quaternary structure of it is formed of 3 long polypeptide chains or strands, and it's twisted to form a rope, and it gives rigidity, and it's usually insoluble in water. So the examples of secondary structures would be silk made from silkworms and cobwebs made from spiders. The examples for tertiary would be any enzyme. The examples for quaternary proteinous structures would be hemoglobin and collagen. Of course, there are plenty of more examples, but those were just some basic ones so you can remember. Now, proteins can be categorized into 2 different categories: fibrous proteins and globular proteins. So, fibrous proteins consist of parallel polypeptide chains cross-linked at intervals to form long fibers or sheets, kind of like the beta-pleated proteins. The globular protein chains are tightly folded to form spherical shapes. The fibrous is linearly arranged, and the globular proteins are, well, globular. The fibrous proteins are more stable than the globular proteins, and they are also insoluble in water. Now remember that the globular proteins are generally soluble. The fibrous proteins are designed for a structural role. The globular proteins are designed for a non-structural role, which means for metabolic activities. The examples for fibrous proteins would be collagen and silk and keratin. Collagen is a major constituent of connective tissues and tendons and bones. Silk is generally used for stuff like clothes. And keratin is a component of your hair. Now don't mix up keratin and chitin. Chitin is found in the exoskeletons of arthropods. The examples for globular proteins are enzymes, antibodies, and several hormones. Now we know how a protein is formed. A protein is formed by the conjunction or the union of several amino acids. But what causes a protein to break? If the bonds holding the shape of a protein are broken, it is called the denaturing or the denaturation of proteins. You have to remember that the primary structure of the protein still remain the same because disulfide bonds are generally strong and they do not break easily. To be considered a denatured protein, it's generally from the loss of a specific 3-dimensional conformation or a specific 3-dimensional function. So it's generally for more complex protein molecules. Single cells of proteins don't break. So the loss of 3-dimensional conformation of molecules and the loss of a biological function occur during denaturing, and remember that this process is irreversible. They usually ask a question in the difference between the DNA strands and the strands of protein. The main point that they're looking for is saying that The denaturing of proteins are irreversible, and the denaturing of DNA is reversible because DNA does get back together, and that's why DNA makes an awesome molecule to carry genetic information. So what causes the protein molecules to break or denature? It could be a lot of factors actually. There are around 5 or 6, but here we go. One is heat. If the temperature goes above 40 degrees Celsius, it passes the optimum temperature for metabolic activities in the body and the hydrogen bonds start to break. If it's exposed to strong acids, it could break, and it could also break if it's exposed to strong alkali or bases. Inorganic chemicals like cyanide or organic chemicals can break them. And finally, mechanical forces. If it's applied, it can break the hydrogen bonds. So that covers what exactly proteins are. It's not as long as the description of carbohydrates because carbohydrates were much more complex. However, the uses of proteins, it has a bit more information than those of carbohydrates. So let's go into it. There are 8 uses of proteins, or 8 functions of proteins, and we're going to go down to all 8 of them. I'm going to give you the functions and an example for each. You're going to have to remember pretty much everything I say from here to the end. And, well, you're going to have to remember the stuff I said before as well. That's how exams go. They expect you to memorize everything. The first thing is transport. For example, take hemoglobin and myoglobin. Hemoglobin transports oxygen and carbon dioxide in blood. Myoglobin transports oxygen in muscles. Now you need to remember that myo means muscle. In myosin, myoglobin, myogenic contractions, this thing will pop up again and again, and it'll get easier for you to understand once you get into biology. Once you start learning about everything, it'll start fitting into place. The structural functions of proteins are collagen. It makes up tendons and ligaments like we talked about in fibrous proteins, and keratin, hair, nails, horns of animals, and feathers. So remember that structural proteins are always fibrous. You don't find any globular structural proteins. If you did, however, it wouldn't really serve its purpose. Enzymes are globular proteins that catalyze metabolic acids. Remember we talked about enzymes. All enzymes are tertiary proteins. The examples could be pepsin, renin, chymotrypsin, any enzyme. You'll learn a lot about enzymes, so don't worry about that. So let's just give an example as pepsin. Pepsin hydrolyzes proteins. Then we have storage. Albumin is a storage protein that forms the white of the egg. The egg white is composed of a protein called albumin. Casein is a milk protein, and ferritin is an iron-containing protein. Ferrous, iron. This wordplay gets really easy as things go on. Then we have hormones. They regulate the metabolic activities. An example for the hormones would be insulin. Insulin regulates the glucose level in blood, and glucagon is the counterpart of insulin, which helps keep the level of blood sugar stable by a negative feedback mechanism. Don't worry, there's a whole part about glucose regulation, and we're not there yet. And then we have other hormones like growth hormones, which regulate growth. And some proteins have a protective function. For example, antibodies, they fight infections, and fibrinogen and thrombin, they involve in blood clotting, and it prevents the blood loss. And there are also proteins which are toxic, like snake venom. You don't really have to go into detail about that, but just remember that those kinds of proteins also exist, and they expect you to remember that. And finally, the 8th type of protein is the contractile protein. It is involving in movement. For example, actin and myosin, they are the proteins that enable muscular contraction. So there we have it. Those are the functions of proteins. The functions are transportation, structural functions, enzymes, storage, hormones, protective functions, toxic and contractile functions. When we were discussing about carbohydrates, the tests that we used to identify the carbohydrates were the Benedict's test and the Fehling's test, in which a brick red precipitate was found in the presence of reducing sugars. In proteins, we have two other tests. The tests are the Buret's test and the Millen's test. In the Buret's test, you add 2 milliliters of a proteinous solution, a solution which contains protein, to a test tube and add a equal volume of 5% potassium hydroxide solution and add 2 drops of a 1% copper sulfate solution. A purple color develops if protein is present, and if there is no protein, there would be no change of color. The Millen's test is the other test, and we don't usually use the Millen's test because it deals with mercury, and you can't let kids run around with bottles of mercury, and it's also very expensive. 2 milliliters of protein solution and 1 milliliter of Millen's reagent is boiled together and a white precipitate is formed, and again, as time goes on, it turns into a reddish or a salmon pink color, and that's how you find it. So in proteins, you use Buret's test, which turns into purple, and Millen's test, which turns into pink after going through white. That's different from carbohydrates, which we use Fehling's test and Benedict's test to get a brick red precipitate. And that is it, guys. We have completed proteins. Thank you guys for watching. That is the end of LankanTutor Biology 2.4. Next time we're going to be going into lipids in Biology 2.5, and it's going to be the second final part in organic molecules. If you are not watching this on LankanTutor.com, we do urge you to, as it has some notes that can probably help you out. And don't forget to like the video and tell your friends if this video did help you, and follow us on Facebook if you want to know about the new updates. So until next time, we will see you guys later, and as always, have a great day.